Right. So basic thing is you have already seen. I have given you a brief introduction regarding chromosomes. I'm not going to go into the details of chromosome, its structure and function, because we have already dealt with in the next, in the previous class. So just for the sake of introduction, I'm going to see that, word chromosome has a Greek origin to it, which means color, and soma means body. So they are thread-like structures present in the nucleus of the cell, considered made up of genes basically. And during the somatic division, that is the cell division, the chromosomes divide into the daughter cells. So this is all about the chromosomes. Study of chromosomes and the role in the cell division is known as cytogenetics. So as you can see, just a second. Or if you feel you need an introduction to it again, I will definitely do it. But we have already dealt with it in the last class. So now, please give down. Right. So when it comes to karyotyping means, or a karyotype is the number and the appearance of the chromosomes in the nucleus of a eukaryotic cell. So the complete set of chromosomes, how they are arranged in an organism, the number of it is known as the karyotype. So study of it is known as karyology, and in the standard format, the chromosomes that are present are represented as a karyogram or an ideogram. So that's what karyon means, again, means the nucleus, and ideogram is the karyotype that is represented. And it is like in the chromosomes are arranged according to their size and the position of the centromeres. So if a haploid set of chromosomes in the organism are present and they're arranged in the decreasing size, it is known as an ideogram. So a karyotype in its sense is an ideogram. And when it comes to the chromosome, again, just right now I just kind of told you the basic definition of it. DNA is known as arranged into thread-like structure called as chromosomes. Each chromosome is made up of the DNA, which is tightly coiled. It's only visible during the cell division. Specific number is present in different species for that matter altogether. And the ends of the chromosomes have telomeres to them. Now, telomeres are the tips of the chromosome, and they have an essential role in that it ends the chromosomes, maintains the integrity and the stability of the chromosome, and they have tangent repeats of TTA-GGG sequence in the human beings, and this is maintained by telomerase enzyme, and reduction of this is associated with cell death and cell aging. Now, different species have different number of chromosomes. So, in the case of, it is known as diploid. So, for us, diploid chromosomes, we have 46 chromosomes, which is 2n is equal to 46. Dog has around 78. Fruit fly, that is Drosophila, has around 8 chromosomes. So, that is one of the main important reasons why fruit fly is studied for a lot of, research purposes, in fact. Cat has around 38, and you have different chromosome number in different species given in your textbook. So when it comes to the classification, again, that's what the constricted region of the linear chromosome is known as a centromere, divides the two regions into arms, helps to be kept in an aligned region basically, and it provides an attachment for the provides the site of attachment for the sister chromatids. So this is the centromere, holds the two sister chromatids together. This is the whole of chromosome. The telomeres are usually over here at the ends of the chromosome basically. These are the genes present on the loci on the chromosomes. And these are the two chromatids which are identical in nature. And you have different types of chromosomes classified. We'll just have a look at it. Based on the classification, chromosomes are classified into different categories. That is number, position of the centromere, and the different kinds of groups. So based on the number, they're known as autosomes, and allosomes are sex chromosomes. So autosomes, normally in the human beings, you have 46 chromosomes. So of which 1 to 22 chromosomes are known as autosomes. So 1 to 22 pairs of chromosomes are known as autosomes, and X and Y are known as the allosomes or the sex chromosomes. So they usually are two X's, that is XX is normal female, XY is normal male. And on each chromosome on each member of the sex chromosome is derived from each parent. So somatic cells supposedly have a diploid complement, that is 46 chromosomes, whereas sperm and ova have a haploid complement of 23 chromosomes each from both the parents. So number of the male and the female conceptions are always approximately equal. Now, based on the location of the centromere, they're again classified as metacentric, acrocentric, or submetacentric, acrocentric, and telocentric. So let us just see this. This can also be a dicentric, and this is acentric. That is, you do not have a centromere at all. So in the case of metacentric, centrally located, centromere is present. This is known as the metacentric type. Acrocentric is where the centromere is nearer to the proximal end of the chromosome. So this is a terminally located centromere. In fact, submetacentric chromosome is the chromosome wherein the centromere is somewhat in the middle with an intermediate position. It is not exactly in the middle, but you have it somewhere in the middle. And you have the telocentric chromosome where the centromere is at the tip of the proximal end as a telocentric chromosome. So none of the human chromosomes are telocentric in nature. That is one thing you have to remember. And then acrocentric sometimes, have stalk-like appendages. This ones, have stalk-like appendages which are known as satellites. And these are present during the resting phase interphase cells. They have multiple repeat copies of the genes for ribosomal RNA, that is rRNA. Now, when it comes to the classification, these are dicentric chromosomes that are present. So there are breaks in the chromosomes, then the fusion, because of which it results in two centromeres and the formation of a dicentric chromosome. So as you can see, now depending upon the groups, the chromosomes are classified as groups A to G. Group A is from 1 to 3 and 16 to, that is what there's a slight difference in what is being given in your textbook and over here. So let us follow your textbook. So based on the size, shape, length of the chromosome and stage of the cytogenetic study, the chromosomes have been divided into 7 groups, A to G. So group A chromosomes are 1 to 3, so they are metacentric in nature. Group B is 4 to 5, 4 and 5 for that matter, submetacentric. Please give me a moment, let me check if it all. Yes. Group A, large metacentric chromosome, but chromosome 2 is again submetacentric in nature. So when you see, identification of the chromosomes in your textbook, let us just have that also in mind. The pictures of the individual chromosomes were not exactly present with me. So what is happening is with that you can just follow your textbook. Otherwise, I'll have to take in an ideograms picture. Then you can see over here, right? So please look at this picture and follow what is given in the textbook. That should be helpful for you. So groups A, 1, 2, 3 are largely metacentric in nature. And then you can see in 2, it is somewhat largely submetacentric. And then from groups 4 to 12, it is around 4, 5 is coming under group B. Group C is 6 to 12. And X chromosome also belongs to the group C. And then group D is around from 13 to 15. Group E is from 16 to 18. Group F is 19 and 20. Group G is 21, 22 and Y. That is what is being given over here. So let us follow what is there in your textbook basically. Now they have given the band resolution also, chromosome band resolution. So when it comes to that, they have just given basically the metaphase chromosomes, how they, how the banding is done basically, and how you are identifying the chromosomes. So for that, let us just look into the preparation of the chromosome for karyotyping, and that is when your banding also comes into place. So when you get to see the karyotype, again, same the process of finding a chromosomal characteristic of the cell refers to the analysis of the chromosome, preparation of the karyotypes basically. So objective is to use gene banding landmarks to identify the individual chromosomes and the karyotyping procedure and the descriptions basically. So when it comes to that, what you're going to do is draw the blood, add a few drops of the blood to the and add to the bottle basically to the for the sera culture. Then add phytohemagglutinin, incubation followed by colchicine, that is colchicine treatment is done, then transfer into different tubes through using a fixator for that matter. And then you're going to that's what, before that you're going to centrifuge the cells, adding low salt solution to it so that RBCs and lymphocytes are eliminated. Then whatever is left, you're going to add it onto the staining slide, and then you're going to see with the GMSA slide. So you're going to get G banding resolution basically. So that's what. Classification of banding is based on the GC and AT rich regions and the constitutive heterochromatin region as well. So you have seen the techniques G banding, Q banding. This is going to be coming in the later part of karyotyping. We will come to the chromosome types in a bit. So if you see the karyotype description and the stepwise procedure, the same thing has been mentioned in your textbook in the last page basically. You can actually see karyotype descriptions given, and then the automated karyotypic systems, which in with methods of chromosome analysis. So the stepwise procedure, whatever is there, is almost similar to what has been given in your textbook. So you can just kind of follow it. Collection of blood under sterile conditions, and then adding the few drops of blood to the culture medium, which is usually HAM10, HAM-F10, and or else you can use even RPMI or TC199. They are all culture media. Then the fetal calf serum or the alternating human AV serum can be used. And then you add the phytohemagglutinin, which is a mitogenic agent. Addition is vital so that this mitogen will promote the increased mitosis basically. So the rate of mitosis will be increased. So rate of division gets increased. This phytohemagglutinin is taken from a French bean, which is Phaseolus vulgaris, are common beans that we have at home, chickbukai, or your that's what. And antibiotics are added so that the culture does not get contaminated. And then it is incubated for around 3 days at 37 degrees Celsius. And then you harvest it by adding colchicine. So colchicine treatment is performed, and 2 hours of it, you centrifuge it. So supernatant is discarded. Pellet containing the cells is treated with hypotonic, that is low salt concentration cells, so that chromatids get separated. Then the cells are suspended in a fixative. Then the fixative is basically glacial acetic acid and methanol in the ratio 3:1. Then you have, then after 3 successive changes of the fixative, cells are suspended and then they are stained. Staining, they have given different kinds of staining techniques over here, that is banding techniques. Q is quinacrine, then G is GMSA, NOR staining is the NOR regions that are there on the chromosomes, they are stained, and C is the centromeric staining. So in Q banding, that is quinacrine banding, Casperson was the person who had demonstrated this sort with quinacrine mustard. And then this requires examination of the chromosomes under UV light. So again, same thing, chromosome, quinacrine mustard subjected to UV light, banding pattern. AT rich regions are darkly stained, GC rich regions are lightly stained. So you can see which is heterochromatin region and the euchromatin region. G banding is GIMSA stain again. So the good slice, can be stained for nearly 10 days. It can be seen in normal saline treated with trypsin and for around 10 to 15 seconds. Then immersed in 70% ethanol, stained with GIMSA, 10% GIMSA for around 6 to 10 minutes, and then the microphotograph is taken. Construction of the G-banded karyotype is seen. So you're going to get one picture with the GC-rich regions and the AT-rich regions giving the euchromatin and the heterochromatin. So when it comes to the N banding, that is you have to take the chromosome from the blood sample, air dry it, and then again add 5% of trichloroacetic acid, and then you're going to add the banding 0.1% normal of HCl at 60 degrees Celsius for 30 minutes. Then you're going to get the non-structural, that is structural non-histone protein link region to NOR region, which is banded or which can be seen. So banding pattern in the structural non-histone protein like proteins linked to the NOR region is seen basically. So C banding is wherein it is the centromeric heterochromatin banding, selective staining of the centromeric region and other regions with secondary constrictions takes place. And you can actually see 1, 9, 16 and the long arm of Y chromosomes with this kind of banding basically. And then if the chromosomes are taken and then they are treated with alkali, that is barium hydroxide, then rinsed in deionized water, and then you add a buffer to it for 1 hour, again stained with GIMSA, deionized water, dried and looked or observed under the GIMSA stained, observed under the oil immersion microscope basically. So you get a banding pattern of the heterochromatin region basically. So this heterochromatin is highly repetitive DNA sequences. So this is seen preferentially. So when these are the techniques that are seen. When it comes to chromosome painting, you have this something relatively new, and chromosome painting. You see that hybridization of fluorescently labeled chromosomes are taking place, and it is specific to the fluorescent regions only, and it allows the visualization of individual chromosomes in the metaphase or interphase, or identification of both the numerical and the structural chromosomal aberrations, that is differences in the human pathology, and it is a highly sensitive and specific procedure. So this is how chromosomal painting is taking place. So when do you take, this is the amniocentesis and which is done for the, that is what, where is karyotyping done? So major use of karyotyping is to find out if at all there is any kind of genetic defect. So you're going to take the sample of the amniotic fluid or the chorionic villus sampling basically. So this is how you perform the amniocentesis. So with an ultrasound probe, with an injection, you remove the amniotic fluid and then you centrifuge it. The fetal cells that are present inside are taken, cultured, then chemically analyzed, and then chromosomal or the karyotyping, the staining is done. So this is one procedure for amniocentesis. And chorionic Windlass sampling is also similar, wherein you take the chorionic, the placenta is over here. This is the chorionic villus. With an abdominal scan, you actually take in the chorionic villus, insert it, and extraction of the piece of the chorionic villus, and then karyotype is done. But having said this, this is quite a risky procedure because it is associated with the miscarriage as well as infection to the fetus, and this is for sure. So you've got to be really careful if at all, if there is any kind of necessity only, they are advised to do this. Otherwise, no. So again, benefits and the risks, and when you can do this, and what is the miscarriage risk. In the chorionic villus sample, it's around 1 miscarriage in 100 samples to 1 miscarriage in 200 samples. This is quite a big risk, in fact. So that is there. So what does karyotyping do basically? Detection of chromosomal abnormalities, the genetic disorders, gender identification, which is illegal in India. So please, but then, they look into the genetic disorders and the chromosomal abnormalities. Then if there is any kind of loss or addition of the chromosomes that can be seen, pre-birth diagnosis of genetic diseases in case, you feel that there is a risk for the genetic diseases. Then identification of chromosomal numbers in different organisms, and identification of proper position of the genes in the chromosomes. All of these are the advantages of a karyotype. Again, disadvantages is, if at all there's any kind of only a single abnormality, it cannot be detected by the karyotype. It should be more in number. Technique only allows for diagnosis. Obviously, genetic diseases do not have cures. Tests like amniocentesis or chorionic villus sample testing are quite risky, and it is a stressful period for the mother basically, and it is a time-consuming process. And minor mistake will change the result overall because you're dealing with the chromosomes over here. So anything misidentification also causes an issue. So this is about the karyotyping. We will come back to chromosomes again because I am not yet done with that particular topic. So, right. So when you see here groups like, as I mentioned, groups 1 to 3 are large metacentric chromosomes, groups 4 to 5 are large submetacentric, group C is, which is the medium-sized submetacentric, medium-sized acrocentric, group D that is 13 to 15 chromosome numbers, group E is short submetacentric, and group F is around short metacentric. Group G has 21, 22 and Y, and then short acrocentric, but chromosome Y is again submetacentric in nature. So when you see the analysis, like they have already told you, prenatally is chorionic villi sampling, amniotic fluid and the fetal blood. Postnatally is the peripheral blood, that is lymphocytes or bone marrow or the fibroblasts. Culture and technique, staining is given over here, conventional, G banding, Q banding, R banding. Selective is the C banding and the AG NOR banding. So AG means silver. So silver staining with the wherein you get the nucleolar organizing regions that are stained or banded. So let me see, there should be one more. So you can just have a look at it basically. So in the case of the fundamental features of each chromosome, identification they have given in your textbook basically, wherein, group A is 1 to 3 chromosomes, large submetacentric metacentric chromosomes, lighter area in the P arm, 2 dark bands in the P arm. Chromosome 1 is what I'm talking about, page 43. You can just have a look at it. And then, they have given each and every chromosome and its identification distinctly. What you can do is you can just have a look at it. And then group 2 is, again, chromosome 2 belongs to group A. And you can see 2 prominent dark bands centrally in the Q arm and 2 bands distally separated by 1 light band. So not very clear, in fact, for that matter. We don't have a very good clear picture except in your textbook. So you can just have a look at it. And chromosome 3 again belongs to the group A, metacentric chromosomes, thick hourglass shaped, and distal area is dark, and to that of the light band of Q arm is thicker in fact than its counterpart P arm. Then cap-like structure in the P arm is seen, and few bands in the Q arm are seen. And when it comes to chromosome 4, which is like, group B, very submetacentric chromosomes. I don't have a better picture. Trying to see if there is a better picture for you in that. I doubt I have a better picture. Almost all the pictures or all the like what do you call it, PowerPoints that we have similar information. So whatever I have and whatever I'm showing it to you on the screen, I will be sharing with them. But please keep in mind that most of the information is similar in almost all the PowerPoints. So what you find is relevant, you can just look at it. But all these are basically reference notes only. They are not your actual, what do you call it, answers. So in your examination, I don't want you to only look at the PowerPoints and finish writing from them. Instead, please focus on your textbook. That gives you more information and much more for accurate information for that matter. And at the same time, whatever is there in the textbook, you please answer that in your examination. That will be helpful. Now, when it comes to chromosome 5, it is again very submetacentric. P arm with a single dark band. As you can see, Q arm has thick clitellum-like band, and subterminal dark band is also seen. You can see that in your textbook as well. You have a better picture. Chromosome 6 is again submetacentric, largest of the C group. This is chromosome 6. Window-shaped pale area between two bands is seen in the P. See this one. It is seen in the P arm. Chromosome 7 again belongs to group C, submetacentric. Two distinct bands in the Q arm followed by less intense dark arm bands. And chromosome 8 also has around, it is submetacentric, two prominent bands in the Q arm. First band in the center of the Q arm. And then chromosome 9, if you come to that, you see that it is very submetacentric. Usually one band is seen in the P arm, and it is having two bands also. Inverted V-shaped pale area below the centromere is seen. Two distinct dark bands in the Q area. Chromosome 10, submetacentric in nature, similar to chromosome 8. And then you get to see that P arm has small dark band in the center. Q arm has three distinct dark bands, proximal being dark. Chromosome 11, as you can see, this has a submetacentric arms basically. It is a type submetacentric. P arm has one or two dark bands. Q arm has very large light area followed by a dark band. Then you can see that a Q arm proximal dark band is making it different from that of the chromosome 9. And you have a thicker clitellum distally near to the telomeres. Chromosome 12, submetacentric. P arm is dark central band. Q arm has a dark clitellum of numerous bands, and it is distally away from that of the telomere. Chromosome 13 belongs to group D, acrocentric. P arm with satellites are also present. See, this is the satellite that is present over here. Has several dark bands. P arm has a short arm or satellites of variable size. Q arm is proximal half of the arm light, divided into two by a narrow dark band. And Q arm has three closely spaced dark bands near the telomere. This looks like a thick clitellum. Chromosome 14 also belongs to group T, acrocentric in nature. Then P arm shows a short arm or is full of satellites. Q arm has two large distinguishing dark bands. One proximal, other distal, but they are subterminal in nature. When it comes to this chromosome 15, it is again belonging to group D, acrocentric, P arm with satellites, Q arm with dark proximal half and light distal half. They are shows P arm has satellites, Q arm has two bands. Again, chromosome 16, pale in nature, submetacentric or metacentric. And then you have the Q arm constituting of the heterochromatin, which is basically dark in nature. Two dark bands appear below this heterochromatin strains. Chromosome 17 belongs to group E. It has submetacentric or very submetacentric centromeres. P arm with thin bands shows proximal central band. Q arm has two subterminal bands. Chromosome 18 belongs to group E. Submetacentric chromosome. Size is similar to that of chromosome 17. P arm is light colored. Q arm is cylindrical in shape. Has two characteristic dark bands, one proximal and one distal. Narrow light telomere is hard to distinguish. When it comes to chromosome 19, belongs to group F. Metacentric in nature, both P, Q arms are light in color. Centromere is dark. and it appears like a smaller version of the chromosome 16. So only the centromere is dark in chromosome 19. Chromosome 20 also belongs to group F. Metacentric or small metacentric centromere is present. P arm is dark, Q arm is light, and there are two narrow, less intense dark bands also present over here. It is not exactly distinguishable from chromosome 19 in fact. Chromosome 21 and 22 belong to group G. They have both acrocentric type of centromeres. P-arm in the case of chromosome 21 is possible with satellites. Q-arm is dark in chromosome 21. In chromosome 22, again, the Q-arm is long, dark, and it has a dark central band. X chromosome and the Y chromosome. These are the sex chromosomes or the allosomes. So in the X chromosome, it is like the centromere is submetacentric in position. P-arm is distinct, strong, dark band in the middle. The size and shape of this X chromosome is similar to the chromosome 7. P-arm has a strong dark band in the center. Q-arm has a proximal dark band, which is equally distant from the centromere like that of the P band. Second dark band is also seen, but it is not as dark as the first dark band. Terminal dark band is also seen. The case of Y chromosome, you have, it is a very submetacentric chromosome. That is, the centromere is a very submetacentric type. P-arm is very short or invisible. Q-arm is dark throughout. It is acrocentric in nature or a very submetacentric. Light area closer to the centromere. P-arm is slightly stained. Q-arm heterochromatin is in the distal position and it is stained darkly. Overall, the chromosome size is smaller than that of the chromosome 21 or larger than that of the chromosome 13. So you have the human chromosome nomenclature. This is based upon the ISCN, which is the International System for Human Cytogenetic Nomenclature, which uses this particular kind of nomenclature. So when it comes to the description of the karyotypes, you get to see that the formula for a normal male is given as XY, and for a normal female, it is given as an XX. So extra or a missing chromosome, you add as plus or minus respectively. So if it is trisomy, so there is an extra chromosome, you're going to write it as for chromosome 18. So you're going to write it as 47XY. In the case of male, we are talking about. So plus 18. And in the case of female, you're going to write with monosomy, that is only one chromosome is present on chromosome 22. You're going to write it as XX minus 22. So the chromosome number becomes 2n is equal to 45 and it is on the chromosome 22 location. So because of it, you're going to write XX minus 22. Addition or deletion of a chromosome segment is again denoted by plus or minus. So there is a there is a syndrome called Crude syndrome. So you have the female with that Crude syndrome. It's going to be again the number of chromosomes is going to be 46 XX, but there's a missing piece in the short arm of the chromosome 5. So you're going to write it as 46 XX 5P minus. So in the P arm you have a chromosome segment or a piece missing. So you're going to write it as 46XX 5P minus. And if at all there is a translocation, we will look into it after some time in the same class in fact. So if at all there is a translocation of the gene between chromosomes 14 and 21, you're going to write it as XY. Since the example in your textbook is XY or the male, so XY 46. So you're going to write it as 46 XY translocation. Denote it as T, and in the bracket, there is a translocation between chromosomes 14 and 21. So from the 14 chromosome to 21. So you're going to write this as 14 apostrophe 21. So this is how you're going to denote a karyotype. And again, XY means sex chromosomes. DEL means deletion. DER is derivative. These are the symbols for the chromosome nomenclature basically. DUP means duplication, INS means insertion, INV means inversion. P is the short arm of the chromosome, Q is the long arm in the chromosome, S is for satellite, T is for translocation. Please give me a moment. And T is for translocation. So all of these have been denoted, and this will go on in all of your next coming up chapters for that matter. So when it comes to the chromosomal abnormalities, we are going to see. So there are different changes that happen in the chromosomes. So it can be normal, it can be abnormal. So if at all it is abnormal, resulting in a disorder, so these are known as the chromosomal abnormalities. So in this lesson, that is unit 3, we are going to look at the chromosomal abnormalities. So, and then the types of chromosomal rearrangements that can take place, and the basis for the chromosomal abnormality, and the conditions that are caused because of these chromosomal abnormalities. So how are the first thing is, when you get to see chromosomal abnormalities is one of the most important reasons for all the kind of health problems. Apologies, I had to plug in the charger. My system had to suddenly stop off. So let me again share the screen. Right. Good afternoon. Am I audible?
Yes, ma'am.
Yes, ma'am.
Sure. Right. Thank you. So that's what, what happened was I had to charge, plug in the charger because of which the system just got abruptly shut down. Anyway, so as I was telling, chromosomal abnormalities are one of the main reasons for all the health issues right now that we are dealing with. Diabetes mellitus also, type 1 diabetes mellitus, you can make out, you can actually identify it in the karyotype. So that is also a genetic disorder. We know that, heart issues or your sugar, diabetes and your liver issues certain times, your cancer, certain cancers are all familial in nature. That is, they are coming through. Even genetics plays a role. Of course, health, living a good health will maximum try to see that the genetic aberrations do not come into place. Certain other genetic issues, nobody can do anything. You just have to identify with it and learn to cope with it, because there are no particular cures as such. So please give me a minute. Let me share this screen. Right. So patience is one thing. There are 2 or 3, PowerPoints that I will be sharing. You can just refer to them, because they have got a good information, and whatever you will be able to understand easily, you can just have it as a reference. All right. So right now I will just start teaching. So when it comes to the chromosomes, you know that there are 23 pairs of chromosomes, and then there are 46 chromosomes. So 23 pairs is known as diploid, and if at all it is only 23 pairs except the mature sperm and eggs, you have a haploid condition. So then in case each DNA is actually stretched and laid end to end, you get to see that there are it is around 2 meters long. This 2 meters long is present in each and every cell that you're talking about. So if you just keep on stretching it, it is unimaginable with the billions of cells that we have in our body, and each cell having 2 meters long DNA. So obviously during division, there is some kind of, during the mitosis, certain times what happens is there is a problem in it dividing properly. 99.99% of the time it divides properly. Chromosomal division, that is mitosis, meiosis, takes place perfectly. But it is that 0.111, not 1%, in fact, which gives rise to abnormalities. So it can be of 2 types, that is a change in the number, or it can be a structural change, that is change in the structure of the chromosomes. So let us just see. So cell division in the germ cells, that is egg cells and the sperm cells, results in the formation of the daughter cells, having half the number of the chromosomes as the original cell, that is a total of 23 chromosomes. This reduction is important because the original number of 46 is restored after the fertilization takes place. So the egg cell has only 23 chromosomes. The sperm cell has only 23 chromosomes. Once they are fertilization takes place, then the embryo will have 2n is equal to 46 chromosomes. So in any case, there is an issue over here. That is when you will have a different number of chromosome number coming in, or a change in the structure. So again, they have just mentioned in your chromosomes itself, that kind of the based on the, the chromosomes are classified based on the type of the centromere that is present, and then, the biggest to the smallest chromosomes, and then the banding types again, G, R, C bands, etc. So the banding patterns, you can identify the chromosomes, and then the banding pattern is determined by the chromatin condensation and the specific DNA protein present at different sites of the chromosome. And then the bands are again individually numbered, so that you have, for example, as given in your textbook, look at page number 73. So bands are again individually numbered. Example is 11q23. So that there is band 23 on the Q arm of the chromosome 11. So 11q23 refers to the band which is present on the long arm, that is the Q arm of the chromosome number 11. So convention is that first it has to state the chromosome number followed by the sex chromosome constitution. Nomenclature is again based on this International Consensus Conference. So for example, 46XX normal female, 46XY normal male. Again, deletion means DEL, duplication means DUP, INS means insertion, I means isochromosome, INV means inversion, R means a ring chromosome, T is translocation, TER is the terminal, MAT means maternal origin, PAT means paternal origin. So you have mitochondrial genes and mitochondrial chromosomes that come into place, which are specifically exclusively inherited from the maternal genes or the maternal chromosomes. And then then the plus means additional chromosome, minus means deleted, or the reduction in the number. So convention is that, for example, total chromosome count, sex chromosomes are followed by the notation indicating the chromosomal abnormality that is present. Now, so 46XY DEL means deletion 22. So there is a deletion on chromosome number 22 and a breakpoint is present at band Q21. 46XX inversion 7 P11Q22. So what happens is XX. So it's a female having 46 chromosomes, but there is an inversion on the chromosome 7 with breakpoints at bands P11, that is the 11, and the Q arm 22. 46XX T 1:6, 1'6, and in another bracket P23'Q21. This means it is a female with 46 chromosomes, but there is a translocation between chromosomes 1 and 6, and there are breakpoints at band P23 on the short arm of chromosome 1 and at band Q21 on the long arm of chromosome 6. And the last example that's been quoted in your textbook is 47XX plus 21. So there's an extra copy of chromosome on the extra copy of chromosome on the chromosome 21, that is trisomy 21 or the Down syndrome. XXX is how you it is usually seen. So G banding, fluorescence in situ hybridization, all of them help in identifying the chromosomal abnormalities that are present. And inheritance also, if you see, all the abnormalities either have a loss or the gain of the chromosome materials. And then you get to see that most of the chromosomal abnormalities are sporadic in nature, and it is small and negligible risk of recurrence that is occurring. And of those are the familial type, that is risk of recurrence is usually less than 50%. And in case of single gene disorders, if there's only one genetic disorder present basically, then that is, there's either a mutation taking place or change within the DNA sequence that can, errors occur within the gene. They can result in absent or deficient or abnormal protein products basically. You can get to see this kind of once in many kind of glycosomal disorders or carbohydrate disorders for that matter. Then the chromosomes of the karyotype with single gene disorder, they're usually normal in nature, except when there is a recessive. Both the parents are carriers, then the child will definitely be having or inherits the disease. But otherwise they are normal in nature. So 46XX or 46XY is their karyotype that is seen basically. So chromosome studies are not recommended for such kind of patients having a single gene disorder such as a cystic fibrosis or Duchenne's muscular dystrophy, etc. Now, what is the impact of this chromosomal abnormalities? You get to see that the chromosomes, despite the fact that until and unless we are actually science students, we do not know that chromosomes play an important role in our like day-to-day life, any stage of the life cycle for that matter. Now we are doing or dealing with cytogenetics, we know the importance of the chromosomes. But otherwise, we do not know that it is because of a chromosomal abnormality that a physical manifestation is being able to be seen. So you can actually see wherein chromosomal abnormalities are playing a role. So in the case of preconception, that is, if at all there is a familial history, there are patients with Down syndromes, then in the family already, then you're going to have a doubt whether the newborn, the child who's just been conceived, or whether you'll have to go ahead for a child. That is when you'll actually think about the chromosomal disorders, or the genetic counseling that is going to be taking place. Members of a high risk ethnic groups for that matter, and if there was a previous instead infertility or sterility problem, or exposure to mutagens, those who are dealing with EDTA or colchicine on a day-to-day basis, or who are working in high risk radiation atmosphere for that matter, they will actually have to think and go ahead for genetic counseling as to see whether if at all there is any kind of chromosomal abnormalities. Then if at all the mother has diabetes or epilepsy, and if it is a consanguineous marriage, that is marriage within a family, marriage within the first cousins, and that is there, and anxiety over childbearing. Right now, even mental health is very important, so that you will get to know if at all that plays an important role in your genetic makeup altogether. Then if at all there have been previous miscarriages or pregnancy loss, and if at all there was a stillborn child or any kind of child with a birth defect, that is neural tube defect or Down syndrome for that matter, or any other abnormalities, anomalies. So right now what they're doing is prenatal, that is before, during the pregnancy itself, there are certain diagnostic tests that are being done. I'll come to that. So these are done routinely in the third month of the pregnancy. There is a genetic test that can be done for any kind of anomaly. Otherwise, in the sixth month, you will have the STIFA scan, which is for the fetal anomalies that can be seen. Either the neural tube defect or for the Down syndrome. What is the kind of ratio between the head and the the body length, and all of this can be seen in the TIFA scan for that matter. So other than that, who has to get a genetic or a prenatal tests for chromosomal abnormalities? So those women are who are older than 35 years or older at the time of delivery for that matter, and a woman or a partner who is known to have chromosomal rearrangements or abnormalities, family history of neural tube defect, or previous child having any kind of congenital anomalies or chromosomal abnormalities for that matter, abnormal levels of maternal serum alpha fetoproteins, human HCG or estriol for that matter. All of these actually cause an issue. And women exposed to infectious diseases, radiation, drugs, or other environmental agents during the pregnancy. Such women are recommended or highly advised to go for chromosomal abnormality tests, right? So definitely, I will send the PowerPoints, like I already told you. I had sent 2 PowerPoints last night, and the rest, the second chapter, that is karyotyping, and this chromosomal abnormalities, whatever is there right now on the screen, I will send these anyway. Now, due to the increased risk of the chromosomal abnormalities for the women aged over 34, in fact, all pregnant women who are 35 or more at the time of delivery are given the option of prenatal diagnosis. And serum markers are also seen between 15 to 18 weeks of gestation. And in the case of infancy and childhood, okay, fine. Prenatal over. Now, after the child is born. So, if at all there is a history of intrauterine growth retardation or failure to thrive in the child, abnormal growth patterns, that is short stature, obesity, or there's an excessive growth in the child, and different kind, like different set of genitalia are seen. Early onset of puberty. Usually, puberty is from 12 years and above. If it is early onset, say around 10 years, even 10 is right now considered normal, but unfortunately, it is not, because of the kind of heat that we are experiencing all over, like the increased heat. The puberty age has actually come down to around 11, 10, 11. Earlier it was 12 to 13. Anyway, so you have microcephaly, macrocephaly, craniosynostosis, which is like, the head structure is different. Microcephaly means the head is very small. Macro means enlarged head. So then if at all there is any kind of delay in the motor skills of the child, or the language skills, soft skills for that matter, or there is any kind of mental retardation, hypotonia, that is in swelling basically, and parents or siblings or other relatives having such kind of similar issues, facial features which are not normal, and if at all there is any kind of abnormal body or limb, asymmetry or congenital anomalies, and cleft lip for that matter is one type, and a significant regression in the developmental progress, unusual behavior. For example, minor malfunctions like hand biting, hand flapping, autistic symptoms. So autism is again, a whole spectrum. It is known as autism spectrum disorder. So such kind of children, you can see they're physically completely normal looking, in fact. But then it is the mental development which is actually delayed. So it is a whole spectrum. They can either be severely autistic, or they can be normal, like children who are able to talk with just a little bit of therapy. They can be brought back to our so-called normal atmosphere around, like in our normal schools for that matter, and with basic developmental issues for that matter, or behavioral issues. Or if you get to see abnormal sleep pattern, a child has to sleep for more than around 13 hours, at least up to the age of 2 or 3. It's the bare minimum, 13 to 14 hours. If the child is not able to sleep for such a long time, or if the child is sleeping for more than that time, then there is an anomaly or abnormal sleep pattern is seen, because of which that is also a type wherein you will have to be a little careful and look at the scenario as to what is causing it. And all this is when you have to go for the chromosomal abnormality, the tests basically. And in the case of adolescence and adulthood, if at all there is any kind of abnormal sexual maturation, amenorrhea, that is, there is no menstrual cycle at all in the case of females, or if there is a delayed puberty, growth retardation, excessive tall stature, can be hyperthyroidism or hypothyroidism for that matter, members of high risk groups. So they can all go for the chromosome abnormalities or genetic testing for that matter. Now, when it comes to the chromosomal abnormalities, you get to see that the numerical abnormalities and structural abnormalities are the two different kinds of aberrations or abnormalities. So if at all there is a difference in or abnormality in the chromosome number, you can get to see that it is either haploid in nature, diploid in aneuploid in nature, monosomy. Let me see if at all I have that. Yes. So haploid is the number of chromosomes in the mature egg and sperm. So it can be just like 23 or N. Diploid is number of chromosomes following fertilization, that is 46 or 2N. So if at all there is on occasion a triploid kid is born, it is going to be 69 or 3N. Tetraploid 92 or 4N. These are very rare and the fetus will not survive. So you will have a miscarriage or an abortion in this particular case. Aneuploidy is the term wherein you see that there is either more than the 46 chromosomes or less than the 46 chromosomes. Most common form of aneuploidy is hyperploidy, wherein you have trisomy, that is 2n is equal to 46 plus 1. So you have 47. Or tetrasomy, which is 2n is equal to 48. So 2n plus 2, 46 plus 2, 48. So over here, most trisomy features are usually spontaneously aborted. That is, there is a miscarriage, except in the case of trisomy 21, that is Down syndrome, or trisomy 13, and that is Patau syndrome, trisomy 18, that is Edwards syndrome, and if at all there is a trisomy in the case of sex chromosomes, that is XXX female or XXY male or XXY Klinefelter male, these come to term. That is, all the 9 months of gestation is over and a baby is born with this kind of aneuploid condition. Rest of the trisomy fetuses are spontaneously aborted. Another form of aneuploidy is monosomy. So there is only one single copy. So that is known as the, for example, monosomy X is known as the, Turner syndrome. And there are also, again, majority of the fetuses with 45, that is X, are simultaneously aborted, 95% of them. Only few of them come to term, that is the 9 months and the baby is formed or born. Aneuploidy is because of different issues such as non-disjunction or failure of the normal separation of this chromosome pair. So because of which you have aneuploidy occurring. So what happens is, normally the 46 chromosomes are copied, that is replication of the chromosomes taking place. Pairing of them are done. Separation or segregation of the chromosomes during the meiosis is taking place. During meiosis 2, second division of the chromosomes occurs, resulting in the formation of 4 sperms. So one egg with 3 polar bodies, each with 23 chromosomes. So normal situation, the mature eggs and the sperms have only 1 copy of each chromosomes. So this leads to 2 copies after the fertilization. So non-disjunction, it occurs in meiosis 1 or 2, leads to more than 1 copy or more than 2 copies. So again, so they have given different kinds of examples because of that. Let us just have a look at it. Monosomy is right. Loss of single chromosomes, Turner syndrome, that is XO. So loss of the sex chromosome X, non-disjunction. So one gamete receives 2 copies of the homologous chromosome. Other will not have any kind of copy at all. So again, trisomy 13 trisomy 13 is one of the examples that is given in your textbook, and then trisomy 18 is given, trisomy 21, and then the Klinefelter syndrome. Again, this incidence of trisomy 13 is, let me just check if at all there is another right, I'll have to go with this only. Pausing the share for a minute. Right. As you can see, trisomy 13 has, it is like, has an incidence of around 1 in 5,000, and 44% of these newborns which are born with trisomy 13 are actually, they do not live more than the first 1 month, or they more than they die within the first month of their life, in fact. And 69% of the uh, uh, children born with trisomy 13 are dead by the 6 months. Only around 18% of the babies with trisomy 13 survive the first year. And again, there are a lot of disjunction, clinical issues with the child, that it is so difficult for the child to survive. So you have microcephaly, that is small head, scalp defects, small eyes. Then you have extra fingers, congenital heart defects, urogenital defects, and then profound mental retardation. So all of these other symptoms for trisomy 13. Edwards syndrome, that is trisomy 18, is seen in 1 in 3,000 newborns. So it's a ratio of 3:1 projection of females to males. This is seen 30% die within the first month, 50% within the next 2 months, 90% of the children with 18 trisomy 18 or the Edwards syndrome die within the first 1 year. Severe mental retardation, microcephaly, prominent occipits, the chin is not proper, short sternum, clenched fists with overlapping fingers, hypertonic neurologically, kidney, heart, GI abnormalities, all of these are seen in Edwards syndrome. Down syndrome is trisomy 21. 1 in 660 children have the chance of getting trisomy 21. Most common chromosomal abnormality, you have short front to back measurement of the head, flat facial profile, mental retardation, moderate mental retardation, obstruction of the small intestines, and then you have short fingers. They are short in stature. All of these are seen in the Down syndrome children. And 95% of the individuals with Down syndrome have trisomy 21, that is 47XX plus 21. And 4 to 5% have a chromosomal rearrangement. And it is further discussed during even chromosomal rearrangements, because it is seen even for chromosomal rearrangements are seen in such kind of children also. Another one is Turner syndrome, that is it has an incidence of around 1 in 5,000 newborns. So loss of an X chromosome is taking place, so XO. So clinical manifestation, again, short stature and no menstrual cycle at all, infertility, webbed neck. So that is what is all about Turner syndrome. Klinefelter syndrome is 1 in 500 males is the incidence, which is very common again. There is an extra X chromosome for the male. So 47XXY. Presence of this extra chromosome results in failure to masculinize, small testes, sterility, behavioral problems, reaming disability, all of this is seen. So again, they have given a list with the kind of maternal age, risk for Down syndromes, and then total risk for any kind of chromosomal abnormalities after the age 35, especially they have given. See, this is always there. This is the kind of risk. But at the same time, the factor that, having a normal child is also there. So you'll have to count that, go for genetic testing, and then go ahead for children after the age, whatever it is, because they have given right from 20 to 24 onwards. So genetic disorders is something that, cannot be predicted as such. So when it comes to the abnormalities in the chromosome structure, this is the Down syndrome as seen in Lauren Potter from so trisomy 21. Klinefelter syndrome, wherein you have an extra X, X, Y, and XXX syndrome. Turner syndrome is XO. Missing second sex chromosome as such. So XX is not there. This is there in the Turner syndrome. So these are all about the different types of abnormalities. I'll have to go for this particular PowerPoint now. So when it comes to the abnormalities in the chromosome structure, it is because of the chromosome breakage, repair process, wrong segmentation, either translocation or unbalanced rearrangement or a balanced rearrangement. This leads to structural abnormalities such as translocation, inversion, deletion, and duplications. We'll just have a look at it. So translocations, what happens is it involves two non-homologous chromosomes basically. So what happens is from one chromosome, reciprocal is wherein one chromosome, the genetic material goes on to the other chromosome, and then this is followed by a break and exchange of the fragments. So because of which there is a chromosome imbalance. Let me see. I think I have a better PowerPoint regarding this. This will, yes. Duplication, translocation is also mentioned. Deletion. Please give me one moment. I'll have to take in the right slide. So again, different kinds of translocations are mentioned over here, as you can see, which is Robertsonian translocation is one, and you have intrachromosomal translocation, intraradial, extraradial, that is intrachromosomal is from one chromosome altogether. There is a shift in the position either from one arm or from another, from one location to another in the same arm. Intraradial is one location to another in the same arm. Extra is from one arm to the other, from P arm to Q arm, for example, or vice versa. Interchromosomal is segment from one chromosome to the other segment, because chromosomes are all having loci and gene loci. Transpositional is one transfer of the chromosomal segment to another in the same chromosome. Reciprocal is exchange of the segments between the two chromosomes. So they have mentioned the kind of the same examples have been given. CB is being transferred over here, and interchromosomal is also being given basically. Non-reciprocal and reciprocal translocation from one location to another in the same arm. From one extraradial is from one arm to the other. So inter is again, they have kind of mentioned. So between chromosome itself, the arms, there is breakage and exchange of the chromosomes. And then you have fraternal, external translocation, intercalation or insertional translocation, and asymmetrical or eniocentric translocation, symmetrical or eucentric translocation. So it can be familial or de novo also. That is, familial translocation is normal translocation taking place. If the translocation is de novo, that is, parents are not exactly translocation carriers, then fetus has around 10% empiric risk for a possible genetic abnormality. So that is a kind of, so that is what they have given, the normal chromosomes, then the translocated chromosomes with the breakpoints as well. So external translocation, segments are exchanged between two non-homologs. These are the homologs. These are the two non-homologs. So exchange of segments is taking place. And insertional translocation, wherein intercalation or insertional translocation is a new sequence is inserted altogether into this. So orange is the donor chromosome, purple is the recipient chromosome. And you have asymmetrical or eniocentric chromosomes also, wherein you have the arrangement of the gene segments or the chromosomes taking place. Or else you have whole arm translocations, which involves transfer of the whole chromosomal arm, the P arm or the Q arm all together. So you have a centric fusion or a dissociation or a tandem fusion in it. Formation of the new fragment over here, as you can see. Breakpoints over here present. Metacentric chromosome position, creating a large metacentric chromosome, then the fragment falls off. Dissociation is also similar to that. And then this is the tandem fusion basically. So, right. So this is all about the translocation. So I will have to go back to the previous slide as such because it is much more easier to understand for you. In the case of Robertsonian translocation, as given in your textbook, there is a breakage of two acrocentric chromosomes, fusion of the long arms, short arms are lost totally. This short arms are lost. So you have the short arms, which they do not have any particular significance because they only have genes for rRNA, ribosomal RNA. So this is seen in chromosome number 45, but there is no loss of genetic material. Therefore, it's a balanced translocation taking place. So in the Down syndrome also, as I have already told you, there is a particular translocation taking place. As you can see, 14 and 21. So you get a normal carrier and a Down syndrome carrier also. And in the case of deletions, you get to see that loss of a part of the chromosome. So deletions of the larger portions of the they are like chromosomes, when they are deleted altogether, they're life is not exactly taking place. It is incompatible with life. So 10 to 15% of the deletions are seen in balanced translocations. Only one parent is around 85 to 90% of it true deletions. So this is wherein loss of the segment of the chromosome is taking place. So for example, in Crude syndrome, so it is seen if it is in the females for that matter. You get to see the number of chromosomes is same, but only the part 5p segment is lost. In the case of Wolff-Hirschhorn syndrome, you have the part 4p which is being lost. And other deletions, you get to see that usually it is seen in the distal end of the short arm, that is a P arm. Other deletions are the deletions of 18p, 18q, 22q, 15q, etc. So they are clinically severe, and you can get to see the duplications. So these are all the basically micro deletions in nature. And in case of insertions, you get to see that there is a rare non-reciprocal type of translocation wherein a chromosome gets inserted into another chromosome. Such inversion is wherein what happens is with the breakage of the chromosome, rearrangement is taking place, because of which a segment is reversed. So there is a centromere involved and paracentric only if an arm is involved. I request you all to please mute yourself. And in the case of ring chromosomes, there is a breakage occurring. This is another structure anomaly that is taking place. And there is a break in each arm, and both the sticky ends join together, forming a ring chromosome. The distal fragments are totally lost, and this is seen in around 1/5 cases of the Turner syndrome patients. So, and in the case of a duplication wherein a extra chromosomal segment within the same homologous chromosome is added, and then it is added to the non-homologous chromosome. Again, it is dependent upon the kind of segment that is being added. You also have a Philadelphia chromosome wherein there is a translocation between the chromosomes 22 and 9 on the long arm. And there is a shorter arm of the chromosome 22 called as a Philadelphia chromosome. This is seen in chronic myelogenous leukemia. So it is easy to identify such kind of patients. If at all they have a Philadelphia chromosome, that they are susceptible to myelogenous leukemia. It indicates the outcome better. So it is you can treat the leukemia much more faster. You also have a fragile X syndrome wherein it is caused by XY sex chromosomes complement to the karyotype having the with the 46 chromosomes. No structural abnormalities is seen, but at the tip of the X chromosome of the XY, you can see the kind of fragile X that is there basically. So again, not all cells might be showing such kind of an fragile X cell basically, or a chromosome basically. It is displayed only in up to 5 to 50% of the cells. So cytogenetic tests are not being used currently for determining if at all there is a fragile X in the chromosomes. So this fragile X syndrome causes developmental delays and also attention deficit disorders or ADD. And again, if at all there is any kind of common chromosomal abnormalities, what are the basic features that are seen? So we have already read about it. I will kind of send this particular, what do you call it, uh, the PowerPoint. Anyway, when it comes to the dysmorphology, how do you assess if at all a child has any kind of genetic disorder? So you can get to see that the body proportion, that is the length of the arms, legs basically related to the trunk, head versus trunk ratio, length of the arms and legs compared to the trunk, achondroplasia, or for that matter, how the growth of the head is taking place, whether microcephaly or macrocephaly, and that is seen. Or if at all there is any kind of achondroplasia, that is absence of cartilage during the growth, so this results in dwarfism. Or if at all there is excess growth, that is gigantism. So then dwarfism is recognized with club feet, cauliflower ears, hitchhiker thumbs, and it is again autosomal recessive disorder. Give me 5 minutes. I will just finish this particular chapter. Almost done. You have around 2 pages according to your textbook. See, there is a whole chapter and there is a whole PowerPoint regarding this, but I'm not really sure because the thing is it is very graphic in nature. So just looking at it, if you want, I will share it with you. Otherwise, no. As of now, I'll just kind of show if it is helpful for you. In case you want, I will always be able to share. Just give me a moment. So head and senior, microcephaly is one, macrocephaly is one. The facial features are different basically. Hydroencephaly is when there is a lot of dilatation of the ventricles over here, as you can see. Then you have malformations in the head. Then this is the skull and malformations of the same. Then you have the neural tube defects, that is asencephaly or cranioceresis. So as you can see, it's extremely, and this is spina bifida. So facial features make an prominent, as you can see, microcephaly or macrocephaly. Fetal alcohol syndrome also has this kind of facial features which are different for that matter from the normal ones. Differences in the hands and the feet, short fingers, short length basically. So floppy infant is seen basically. Then congenital parietal foramina. So the sutures that are present over here, you have a hole here, because of which you have the structure of the head which is different. This is brachycephaly. I think it's given in your textbook. You can always have a look at it. And then you have different kinds of segments. You have a conical head over here. Plagiocephaly is oblique shaped skull. So that's what different kinds of genetic disorders giving different kind of output as such. When it comes to the ears, you have different small, large, normal ears, tags, malformed ears, all of that. Then hypoplasia of the pinna. Pinna is the earlobes. Then that's what eyes, again you have abnormalities as seen over here, and you can get to see hypertelorism, or for that matter, then before and after. And these are the kind of genetic defects that you can see: congenital glaucoma or cataract for that matter. Down syndrome eyes are different basically. Then when it comes to skin and hair abnormalities also, nose also there's a difference, in the nose you have nasal bridge which is depressed, cleft nose deformities, peaked nose. These are all genetic again in nature. So when it comes to the mouth also, you have different kinds of abnormalities that can be seen. So what I'm going to do is I'm going to skip a bit, and you can see the hand abnormalities again: polydactyly, syndactyly. This is incomplete syndactyly, brachydactyly, as you can see. Then camtodactyly, genetic disorders again, clenched hand. Acaria, Macrodactyly, one finger is enlarged, foot, different kinds of feet. Then absence of patella, that is kneecap basically, and absence of the entire limb structures. Then abnormalities of the spine is seen, abnormal as in the case of asencephaly. This has been mentioned and the pictures is there for you in the first thing. Then say shape, size, symmetry of the chest bone is also important over here. Abdomen also defects if at all there is any kind, it is seen basically. And then you can see that the abdomen shows abdominal wall defects. Then dilated umbilical cord ring, that is also there actually in the pictures. I just skipped through them in fact. And then if the genitalia, definitely male or female, large testes are seen in male with Fragile X syndrome. Small testes are seen in Prader-Willi syndrome. Developed delay in the secondary sexual characteristics in both Turner syndrome and Klinefelter syndrome. And ambiguous genitalia is seen in different genetic disorders again. So a lot of children born with genetic disorders have a lot of physical malformations and mental disorders. So syndrome means a group of major and minor abnormalities. Malformation is a delayed malformation of a particular organ or an organ system. It can be seen in caused by a group of genes or one single gene altogether. Deformation is in the case of where there is a breech position during the delivery. This causes a congenital dislocation of the hip. So because of which you get a breech head, that is the elongated head. So it can be corrected as such. And disruption is breakage of the normally formed tissue or the structures for that matter. So limbs can be amputated by an amniotic band for that matter. And if at all there are any cysts, they can be removed. So when it comes to the dysmorphology and chromosomal databases, you can see there are different kinds of databases with the different kinds of genetic disorders mentioned. One of the most common ones is the OMIM, that is Online Mendelian Inheritance in Man. The disorder that is there, where is it, on what part of the gene, what part of the chromosome, is it a long arm, the short arm, what kind of disorder, genetic aberration, the chromosomal aberration, all of it can be mentioned, and the physical manifestation of it, and what is the result of it. All of this can be seen in these databases. So OMIM is one, London Dysmorphology Database is one, chromosomal variations in man is one. So to summarize this lesson, we get to see that 46 chromosomes, 23 pairs, and chromosomes 1 to 22 are known as autosomes, and they're present in both males and females. 23rd pair is the sex chromosome, differs in both the sexes. Females have 2 copies of the X chromosomes. Male has 1 X chromosome from the mother, Y chromosome from the father. And chromosome analysis, requires the cells to be cultured and then harvested. Then the karyotype of the chromosomes, based on the size, shape, and banding pattern can be seen. And routine chromosomal analysis or the stains are done basically. And then GMSA staining or G banding is very common basically. And then you get to see that fluorescence hybridization studies, fish studies are also done to identify the microdeletions or if at all there is any extra chromosomal material present. And at fertilization, total chromosome count should be 46 and it is restored. Following mitosis, all the somatic cells have 46 chromosomes. Non-disjunction results in aneuploidy. And then the chromosomally abnormal genes are fertilized, resulting in, if at all, the fetus gets miscarried, or it will come to term in certain cases, such as the trisomy 21 or the Down syndrome. When these chromosomally abnormal gametes are fertilized, then the incidence of non-disjunction increases with maternal age. So early 20s, 1 in 500 chance of having a child with a chromosome abnormality. Increases in 40s to 1 in 21. So where is 1 in 500? Where is 1 in 21? Abnormalities in the chromosome structure occur when 1 or more chromosomes break. And during the repair process, the broken ends of the chromosomes are rejoined incorrectly. So the balanced chromosome rearrangements are physically and intellectually normal. But if at all there is any kind of imbalances, then you have chromosomally abnormal gametes that are produced. Then this comes to clinical attention following the birth of the child with a chromosomal abnormality. Then they are either likely to have a miscarriage or can be identified if at all there is a chromosomal study that is done to determine if at all the loss of the pregnancy. So this is all about your what do you call it, chromosome and its abnormalities. We are done. Tomorrow we are going to see the genetic disorders. All right. Thank you so much.
Thank you, ma'am.
Thank you.
Thank you. May I know your name, please? Hello.